So, good afternoon, everyone. Today, we'll be discussing about the bacterial infections and how they mediate an immune response in the body with an example of tuberculosis. Okay, so I'm Rishabh, and we'll start with week 11 of the host pathogen interaction course by Professor Himanshu. So, moving on, the bacterial infection, that means whether any bacteria is infecting the body. Okay, so there can be either intracellular bacteria that affects that stays inside the cell. That means either Mycobacterium tuberculosis or Listeria, or there can be extracellular pathogens that remain extracellularly, that are Salmonella and Shigella. Now, route of entry, there are different route of entry that every bacteria follows. First is the broken skin, that means Clostridium tetani, that causes tetanus, that is enters through the body through the broken skin. Then we have the respiratory tract, that is the Mycobacterium tuberculosis. Then GI tract, that is Salmonella and Shigella. So GI tract infections are mainly caused by Salmonella and Shigella. Then we have genitourinary tract, so that is Neisseria gonorrhoeae, that causes the gonorrhea. So that is a type of the STD. So these are the different routes of entry of different bacteria in different conditions. So infection to the host and disease outcome depends upon the inoculum size. Inoculum size means the number of bacteria infecting the body. Virulence of the bacteria, how virulent the bacteria is, then host immunity to clear the infection. That is the main. The immune the main infection process involves the attachment or adherence to the cells, mainly the epithelial cells, and then how they enter and multiply and spread inside the body in different tissues via the lymphatic system to the bloodstream. So, to enter the bloodstream, every pathogen follows the lymphatic system. Okay, the immunity against bacterial infection, how does the human body actually cope up with the bacterial infection? So, in the innate immunity against bacterial infection is the humoral components. That means antimicrobial peptides such as MBL, mannose binding lactone, uh, and the mannose binding lectin and the complement system. So, this is the part of the innate immunity in which the complement system basically helps in uh, preventing the bacterial infection. Then, the in case of the cellular components of the innate immunity, we have the PRRs. For example, TLRs and NLRs are basically helpful in preventing or recognizing which bacteria has infected the body and developing immunity against that. Okay, then for the extracellular bacterial infection, antibodies and the Th2 immunity. So the T helper second cohort in immunity for extracellular bacterial pathogens via the complement activation, phagocytosis via opsonization, and neutralization of the bacterial toxins. Okay, so this is the case with the adaptive immunity in case of the extracellular bacteria. For the extracellular bacterial infection, we have also the Th1 immunity. Okay, so this was the about the Th2 and the antibody immunity, and this was about the Th1 immunity. Okay, then moving on to the genomics and the bacterial pathogenicity. So how different components in the bacteria actually helps in the bacterial pathogenicity. So we have the clonal nature of the bacterial pathogen. There are many types of influenza. So Haemophilus influenza, but the clonal Haemophilus influenza type B is commonly associated with the disease. So there can be different clonal population of the bacteria that causes the disease. Okay, then we have the mobile genetic elements. The primary mechanism for exchange of genetic information between bacteria include natural transformation, or transmissible mobile genetic elements like plasmids, transposons, and bacteriophages. So, how do a bacteria actually transmit the infection through other bacteria is the presence of the mobile genetic elements. So, for example, it has plasmids. So, plasmids are extracellular DNA that carry some special genes in terms of providing the virulence of the bacteria. Okay, then we have the transposome or the jumping genes. So these genes basically that have the infectious properties travel from one bacteria to the other. And then the bacteriophages, those are the viruses that kill the bacteria but can actually contain some of their genome parts that carry the infectious genes. Okay, so this is how the mobile genetic elements help the bacteria to increase their pathogenicity. Then pathogenicity islands, that is large group of genes that are associated with pathogenicity are located on the bacterial chromosome are termed as pathogenicity islands. So what happens in case of the bacteria, most of the genes that are present in the form of operons. Okay, so all the genes that have related function or present in a pathway, they are present in the form of operons. Okay, so now these operons basically helps in, so these operons are for the pathogenicity of particular bacteria are present in the bacterial genome. Okay, so those help in increasing the virulence of that particular bacteria. So moving on to the different examples of the virulence factors included by the genes on the mobile genetic elements. So we have, for example, these are the examples. For example, in E. coli, we have heat labile and heat stable enterotoxins that cause diarrhea. Then we have hemolysin, that is cytotoxin of invasive disease and urinary tract infection. Then, in case of E. coli and Shigella species, we have adherence factors and gene products involved in mucosal invasion. So there are different virulence factors in different bacteria for different infections. For example, in E. coli, we have enterotoxins that are heat labile and heat stable that cause diarrhea. Then we have hemolysin that causes urinary tract infections, and then we have the mucosal invasion gene products and adherence factors. Then we have, in case of Bacillus anthracis that causes anthrax disease, the capsule is essential for virulence and that is present on the plasmid. The edema factor, lethal factor, and protective antigen are all essential for the virulence, and that is present on the other plasmid. So it contains two plasmids. Then this was the plasmid encoded genes that help these bacteria to actually infect. Then we have phage encoded genes. So the Clostridium botulinum, these contains botulin toxin that causes paralysis, and these are encoded by a phage. Then we have Corynebacterium bacterium diphtheria. So it contains the diphtheria toxin that inhibits protein synthesis in humans. Then we have Vibrio cholerae. So cholera toxin that can cause a severe watery diarrhea. Okay, so these all genes are present. These are present on the phage. So these are encoded by a phage. So that was actually being infected, that infected the bacteria containing these virulence genes. Okay, then we have the example of pathogenicity island that are present for of human pathogens. So we have E. coli. So pathogenicity island are named as PAI. Okay, so for example, in E. coli we have S36. So these are alpha hemolysin fimbriae adhesions in urinary tract infection. And we have another PAI J96. So these are also alpha hemolysin P pilus So in urinary tract infection. Then we have E. coli again, O157, that the macrophage toxin of the enterohemorrhagic E. coli. Salmonella typhi SP1, so invasion and damage of the host cells for the diarrhea. Then we have Yersinia pestis, HPI, and or the PGM. These genes that enhance iron uptake. Vibrio cholerae EL TOR O1, so VPI1, that have the neuraminidase utilization of the amino sugar. Staphylococcus aureus, SCC-MEC. So this encodes for methicillin and other antibiotic resistant genes. Then we have Staphylococcus aureus, that is SAPIL1, toxic shock syndrome, toxin 1 and enterotoxin. Then we have Enterococcus faecalis, NPM, cytolysine biofilm formation. So these are the different bacterial species that have the pathogenicity islands. So these are the different full forms of the terms that have been mentioned here. Okay, now moving on to the host immune responses to bacterial infections and bacterial evasion mechanisms. So the infection process, the attachment to the host cell, blockage of attachment by secretory IgA molecules. So these attachment to the host cell will be decreased by the blockage of attachment by the secretory IgA molecule antibodies. So in they help in the bacterial evasion mechanism include secretion of proteases that cleave the secretory IgA dimers, that happens in Neisseria meningitis, gonorrhea, and influenza. Sorry, Haemophilus influenza. Then proliferation. So the host defense include phagocytosis using antibody and C3b mediated opsonization, or the complement mediated lysis and localized inflammatory response. So these are overcome using production of surface structures like polysaccharide capsule, M protein, fibrin coat that inhibits phagocytic cells, and the mechanism in the surviving within the phagocytic cells. Then induction of apoptosis in macrophages, for example, that happens in Shigella flexneri. Then we have generalized resistance of gram-positive bacteria to complement mediated lysis, insertion of membrane attack complex prevented by long side chain in cell wall LPS in some of the gram-negative bacteria. Okay, then we have invasion of the host tissues by mediated agglutination, but there is secretion of elastases that inactivates C3 and C5 as the part of the complement system. So that happens in Pseudomonas. Then we have toxin-induced damage to host cell. So that means neutralization of the toxin by antibody and secretion of hyaluronidase, which enhances the bacterial invasiveness. Okay, so these are some of the mechanisms through which the bacteria invades the humans, and what is its host defense, followed by the bacterial evasion mechanisms. Then we'll move on to the toxins that are produced by bacteria. So there are two types of toxins that are produced by bacteria: exotoxins and the endotoxins, through which it actually carries out its infection process. So the exotoxins include the excreted by living cell. They are high concentration in the liquid medium, while endotoxins are the integral part of the cell wall. In gram-negative bacteria, these are released on bacterial death and in part during growth. May not need to be released to have biologic activity. The exotoxins are released by both gram-positive and gram-negative bacteria, but endotoxins are only found in gram-negative bacteria. The polypeptides of exotoxins is molecular weight 10 kilodalton to 90 kilodalton. Sorry, 900 kilodalton. While the endotoxins are generally lipopolysaccharide complexes, lipidae portion probably responsible for the toxicity. The exotoxins include relatively unstable toxicity, often offered the toxicity often destroyed rapidly by heating at temperatures above 60 degrees Celsius. These are highly antigenic, stimulate formation of high titer antitoxin, neutralizes toxin. Okay, then these are relatively stable, withstand heating at temperature above 60 degrees Celsius for hours without loss of toxicity, and these are weakly immunogenic antibodies or antitoxins. These and protective relationship between antibody titers and protection from disease is less clear than with exotoxins. Converted to antigenic neurotoxins, toxides by formalin, acid, heat, and so on. Toxides are used to immunize less, example tetanus toxide, not converted to toxides. So the endotoxins are not converted to toxins at all. These are highly toxic, fatal to animals or microorganisms, microgram quantities or less. Then it is moderately toxic, fatal for animals in 10 to 100 of micrograms. So the amount or concentration of endotoxin released for needed for its toxicity is high as compared to the exotoxins. Then the exotoxins usually bind to specific receptors on cell, but the receptors in case of the endotoxins are not found on the cells. Exotoxins do not produce fever in the host cell, but the endotoxins produce fever in the host by releasing IL-1 and other mediators. Frequently controlled by the extrachromosomal genes, for example, plasmids. So, all the exotoxins are controlled by the extracellular or extrachromosomal genes, and synthesis directed by chromosomal genes in case of the endotoxins. So, these are the major differences between the endo and the exotoxins that can be seen between different bacteria. Okay, now moving on to the major disease. For example, we have the Mycobacterium tuberculosis. So Mycobacterium begin with is a rod-shaped bacilli. They do not form spores. They resist decolonization by acid or alcohol, that is 95% ethanol and 3% HCl. These are acid fast bacilli, hydrophobic nature, and these are resistant to chemical agent because of their hydrophobic nature, and they resist drying. They can survive in dried septum, sputum for a long period of time. Okay, so these are rod-shaped bacteria, as you can see. Okay, and there are different forms of different species of Mycobacterium that causes different diseases. For example, we have Mycobacterium tuberculosis that causes TB, that is tuberculosis. Mycobacterium leprae causes leprosy. Mycobacterium avium intracellularly that affects the immunocompromised people, who, for example, that are suffering from AIDS. And we have Mycobacterium bovis that also causes tuberculosis in cattle and humans. Okay, then we have non-tuberculosis Mycobacteria NTM. Those are other than Mycobacterium tuberculosis and Mycobacterium leprae. For example, Mycobacterium kansasi and Mycobacterium fortuitium. Okay, so the paradigm for human host pathogen adaptation. So the tuberculosis pathogen is known to initiate infection most efficiently in the lower lung through small aerosol droplets that contain only one to three bacteria. Then tuberculosis appears to lack the classical virulence factors. They are host macrophages to transfer host mucosal barriers to sterile sites deep in the body. Host immune evasion, modulation, and exploitation are the trump cards for the pathogen mycobacteria. And then Mycobacterium tuberculosis' dirty little secret is the hydrophobicity. They fly more efficiently in tiny droplet to bypass the innate immune system. So they have this feature that they are hydrophobic, so they can pass through the innate immune system by traveling in a liquid tiny drops. Okay, so The disease that is caused by the Mycobacterium tuberculosis is tuberculosis. So, tuberculosis is a contagious disease and often severe airborne disease caused by the infection of Mycobacterium tuberculosis bacteria. In many low income and middle income countries, TB continues to major cause of morbidity and mortality. Okay, because if the bacteria actually grows in number inside the body, then in that case, so and only the pulmonary TB is contagious. The pulmonary tuberculosis is contagious. Okay, then the history of tuberculosis. So occurs it occurred as an endemic disease among animals, then long before it affected humans. From the time of Hippocrates, tuberculosis was known as phthisis, a term described from wasting away. Existence of tuberculosis in mummies also existed, dating from 2000 to 4000 BC. Then, so many concepts arose in biology that termed the context of inquiry about the nature of the tuberculosis. In 1882, Robert Koch discovered the causative agent that caused an airborne infection, and that was Mycobacterium tuberculosis. So it has a cell envelope. So talking about the Mycobacterium tuberculosis again, it has a cell envelope that has the mycobacterial outer membrane. So it has in the outer membrane it has mycolic acids. Okay, then we have diacyl trehalose. Then we have uh, thiocerol dimycoserosate PDIM. Then we have polyacyl trehalose. Then we have glycolipid. Okay, followed by the outer membrane, we have the AGP, that is arabinogalactone peptidoglycan. So this is a different form of peptidoglycan that occurs naturally in the bacteria. So it has galactosamine residues, arabinogalactin, and the peptidoglycan attached to it. Okay. Followed by this, we have the periplasm. So these are part of the this is the cell wall, this is the outer membrane, then we have the periplasm. So periplasm consists of the uh, lamb, that is lipoarabinomannan and lipomannan. So these are the forms that are present in the periplasmic space, followed by the inner membrane of the mycobacterium. So that contains the phosphatidylethanol phosphatidyl myoinositol, and the diphosphatidyl glycerol. Okay, then there is different forms of monocyte, so diamonocyte and the hexamonocyte. Okay, so these are the different forms of lipid that are originating outwards from the lipid bilayer. Okay, so then talking about its cell wall, so it induces delayed hypersensitivity. So that's why it is called it is used in Frodent's adjuvant as an for Frodent's adjuvant. Unlike most gram-positive bacteria, these bacilli augment their peptidoglycan wall with an extensive array of complex lipidoglycans, which reflects the necessity to resist desiccation in a soil environment. Okay, then the thick waxy cell wall and the ability to catabolize fatty acids seem to be useful for pre-adaptations to a parasitic existence. So these are the modifications that are present on the membrane of the mycobacteria, in which they actually have augmented their peptidoglycan wall that have an array of lipidoglycans, and this actually helps in maintaining a hydrophobic environment. Okay, then these thick walls actually have the ability to catabolize fatty acids, so that actually is a adaptation for a parasitic existence. Okay, now moving on to how do we actually test for a tuberculosis. So we have Montauk's tuberculin skin test. Okay, so in this, what we do, we actually inject a protein purified derivative into skin. So these are the derivative that is injected into the skin, and now how it spreads throughout a circle. Actually, the result depends on the size of the raised heart area or the swelling, and we check the diameter of that particular circle. And then this test can actually be positive in 4 to 6 weeks of their infection of the by the mycobacterium. Okay, so the positive skin test means that the person's body was infected by a tuberculosis bacteria, and additional tests are needed to determine the person that have a latent TB infection or a TB disease. Okay, then the negative test means the body of the person does not react to the test, and the latent TB infection or TB disease is not likely. Okay, so this is how we detect using Montauk's tuberculin skin test. Then moving on to the Nielsen acid fasting. Okay, so what we do is we actually take a blood sample onto a slide and make a smear out of it. Then we stain with the first stain, that is the primary stain, carbofuchsin. Okay, so first chain is the carbofuchsin. So in that case, what happens? The acid fast positive bacteria that reacts to this carbofuchsin appears pink, while the acid fast negative bacteria also appears pink. Now we heat. Okay, so we heat so that the sample can actually be fixed as well as stained with the carbofuchsin. Okay, so in this case also both the bacteria appear pink. Now the step 3 is alcohol. So all the bacteria that have been stained will now be uh, decolorized using alcohol. So the decolorization of the sample with the acid alcohol. So all the membrane components will be washed away using acid alcohol. Okay, then what will happen? All the acid fast positive bacteria that would have retained the stain would actually appear pink, but the acid fast negative bacteria would actually appear colorless. Now we apply methylene blue, so this is a counter stain. Okay, so all the bacteria that are acid fast negative will now be stained with methylene blue. but all the positive bacteria will be stained with carbofuscin. Okay, so this is how the acid fast Zeal Nielsen acid fast stain reaction works. Okay, and we can detect the presence of microbacterium in a serum sample. Then we have IFN gamma release assay. Okay, so what we do, so what does this assay do? So this, in case of the antigen presentation, the cells that are present there, the antigen presenting cells, they encounter an antigen. Presenting cell ingests then digests the antigen, and after incubation it releases the IFN gamma. Okay, so what happens is, now if the plasma cell have the particular mycobacterium antigen. So the T cells that are specific to the mycobacterium will release the IFN gamma after interacting with the antigen. Okay, so this is what the main principle is, and then after that the released IFN gamma can be checked through ELISpot spot assay. Okay, so the T spot TB, that is the ELISA ELIS immunospot, that detects the level of infection that the person holds. Okay, so for example, here we have a T cell. Okay, so this T cell, when encountered with mycobacteria, will produce IFN gamma. So now this IFN gamma will be actually detected detected using an ELISpot assay. So now the number of spots, if increased, that means there is infection with mycobacteria, and if the number of spots decreased, that means the infection is not there. Okay, so this is how this test help in detection of the mycobacterium antigen. Okay, now we'll move on to the pathogenesis of the mycobacterium tuberculosis. So what happens, there are droplets from a diseased patient. So this is a diseased patient, sorry, this is a diseased patient that have a mycobacterium infection. Now the droplets from the coughs and sneezes by an infected person can lead to a non-infected person. Now the mycobacteria actually infect the alveolus. Okay, so the alveoli of the lungs. So these are the macrophages and the monocyte and the dendritic cells that are present on the alveolus. Now this mycobacteria enters the particular epithelial cell by puncturing it. Okay, by puncturing it, then it infects the interstitial macrophages. So the that leads to the formation of granuloma. Okay, so that is a cellular granuloma in which the macrophages and all the immune cells are affected by the mycobacterium bacteria. Now this leads to a necrotic granuloma. So the granuloma is basically the hiding spot for the mycobacteria. Okay, now, these are the different types of cells that can be present in a granuloma. So these include macrophages, epithelial interstitial macrophages, apoptotic macrophages, and so on. And this is a Gantt cell. Okay, so this contains all the cells, immune cells. This contains all the immune cells that carry the infective bacteria. Okay, now moving on the spectrum of TB. So in case of infection eliminated bacteria, what happens? This is a typical test that can be negative or positive in that case. Okay, so in case of the TST, the test will be negative in all the cases when the innate immune response has been activated, or with acquired immune response, that AFN gamma and the TST will be positive, but the other test would be negative. But in case of the latent TB infection, so the mycobacterium has actually been present, hiding in the body. So in that case, the TST and the uh, IFN gamma test is positive, but the culture and the smear will be negative because it is a latent infection. But the therapy is preventive therapy. In case of the subclinical TB disease, all the tests would be positive, but the infectious can be sporadically, symptoms mild or none, and there will be multi-drug therapy that will be given to the patient. In case of the active TB disease, all the diagnostic tests will be positive, while there is mild to severe symptoms, and there will be multi-drug therapy. Although primarily the pathogen is Mycobacterium tuberculosis that cause the disease throughout the body, and it can spread to bloodstream, lymphatic channel, bronchi, and the GI tract. Intracellular site growth happens at monocyte, reticuloendothelial cells, and the giant cells that we saw in the uh, granulum. Okay, so this is how the spectrum of TB is seen and how it gets the treatment. Okay, now let's talk about the some treatment given to the TB patients. Okay, so the first line of drug for TB is isoniazid. Okay, so this is the isoniazid formulation that is given to the TB patients. Then rifampicin that inhibits bacterial DNA dependent RNA polymerases that inhibits the RNA synthesis. Then pyrazinamide that disrupts the membrane transport and the energetics. Then we have ethambutol that is active against multiplying bacilli by interfering in the biosynthesis of the arabinogalactone. Then we have streptomyces that inhibits 16S rRNA of the 30S ribosomal subunit that inhibits with the formyl methionine tRNA, that is the first residue for the protein synthesis. So these are the first line of drugs that are given to a TB patient, followed by the second line of drugs for a TB patient include the potent inhibitor of neuroamidase lipid biosynthesis, that is kanamycin, sapromycin, ethionamide, cyclosporin, ofloxacin, and ciprofloxacin. And for the drug resistant TB, when the drugs are treated to TB are misused or mismanaged, then people they, actually who do not complete the full course of TB treatment, so they tend to acquire multi-drug resistance. Healthcare providers that prescribe the wrong treatment, either the wrong dose or the length of the time. Then drugs for proper treatment are not available, and drugs are of poor quality. So in case of the multi-drug resistant TB that are resistant to at least isoniazid and rifampin, the two most potent TB drugs are um, there. The pre-existing drug-resistant TB is a type of multi-drug resistant TB caused by the TB bacteria that are resistant to isoniazid, rifampin, and fluoroquinolone, or by TB bacteria that are resistant to isoniazid, rifampin, and second-line injectable amikin, cipromycin, and kanamycin. Extensively drug resistant TB is a rare type of MDR, the TB caused by the TB bacteria. So those are resistant to isoniazid, rifampin, fluoroquinolone, and a second line injectable amikane, ceprosine, and kanamycin. And these TB bacteria are resistant to isoniazid, rifampin, or fluoroquinolone, and bedaquiline or linezolid. So these are the multi-drug resistance bacteria that are develop resistance after either due to the wrong treatment or the untimely treatment of the drugs. Okay, so now we'll start with some of the questions related to this. Which of the following statements about endotoxins are true? So the options are: they are found only in the gram-positive bacteria. No, they are found in gram-negative bacteria. They are components of the outer membrane of the gram-negative bacteria. No, they are present inside the gram-negative bacteria. They are stable at high temperatures. Yes. They can trigger fever and inflammatory responses. Yes. They are secreted by live bacteria. Yes. They are unstable at high temperatures. Mm, no. They are converted into toxides? No. They are controlled by the plasmids? No. So the correct answer are 2, 3, and 4. I think instead of 2, fifth would be the correct answer. Okay, so these would be the correct options from endotoxins. Okay, so moving on to the next question: which cells are present in granuloma in a Mycobacterium tuberculosis infection? So what can be the correct answer? So the granuloma basically contains the macrophages, Th1, and the neutrophils. Okay, so the Mycobacterium actually hides itself in neutrophils and macrophages of the alveoli. Okay, so this is how it infects the different tissues in the lungs. Moving on to the next question: which immune response is primarily activated in response to extracellular bacterial infection? So, what can be the correct answer? So the correct answer is option 5. That means both Th2 cell mediated immunity and the complement system helps in recognizing so making our immune response against extracellular bacterial infections. Okay. Moving on to which activity is disrupted by pyranozamide in leading to its anti-tuberculosis activity effect. So what can be the correct answer? So it affects the membrane transport in the bacterial cell membrane. Okay, so disrupting the metabolism of the membrane. Okay, moving on to the next question. The virulence factors of Bacillus anthracis include all of the following except. So what can be the correct answer? So the correct answer is option number 3, that is diphtheria toxin. So this is not a virulence factor for anthracis. This is a virulence factor for the diphtheria, mycobacterium. Okay, so let's move on to the next question. What makes mycobacterium tuberculosis infection a major challenge? So what can be the correct answer? So the major challenge regarding Mycobacterium tuberculosis is the development of the multi-drug resistance. So that complicates the therapy. Okay. So the rest are not related to Mycobacterium tuberculosis infection. Now moving on to the next question. What is the basic difference between the endotoxin and the exotoxin? So what can be the correct answer? So the exotoxins are produced by all bacteria, while endotoxin are only produced in gram-negative bacteria. Then endotoxins are always more harmful than exotoxins. Exotoxins are proteins released into the environment, while endotoxins are part of the bacterial cell wall. There are no significant differences between the two types of the toxins. Both exo and endotoxins have the same mechanism of action. So the correct answer is option 3. The exotoxins are released into the environment, while the endotoxins are part of the cell wall of the bacteria. Okay. Moving on to the next question. What is the limitation of the Mantoux test in the tuberculosis screening? So, what can be the correct answer? So, the correct answer is the cross reactivity with non mycobacterial tuberculosis bacteria can also be shown by this test. Okay, so it will not be able to distinguish between the tuberculosis and the non tuberculosis bacteria. Okay. So moving on to the next question, what does the formation of granulomas in response to tuberculosis infection indicate? So what can be the correct answer? So they indicate a body's attempt to contain the infection and maintain it in a latent stage. Okay. Now moving on to the next question, which of the following statements are true regarding bacterial regulation and pathogenicity? So what can be the correct answer? So the statements are bacteria can exist in both free living and pathogenic states. Yes. Surface properties of bacteria do not influence their adherence to host tissues. Okay. So no, this is false because the properties actually influence that how they will adhere to the host tissues and which tissues it will infect. The environmental signals can trigger changes in bacterial growth modes. Okay, so environmental signals can trigger changes in bacterial growth modes. Yes, the immune system only responds to the presence of exotoxins. No, this is false. It can also respond to the endotoxins as well. Coevolution of bacteria and the host affects infection dynamics. Yes, this is correct because if the immune system is covered with the pathogen, then in that case the pathogen will lose its infectivity with time. All kinds of bacteria can produce endotoxin and exotoxin. No. Secretory IgA plays a role in preventing bacterial colonization. Yes. Then understanding toxin mechanism is irrelevant for vaccine development. No, because it is not a correct option because it will be irrelevant if the toxin mechanism is not known. Okay, so let's move on to the next question. Which of the following are non-tuberculosis bacteria, mycobacteria, NTM? So what can be the correct answer? So option 1 and 2, the Mycobacterium consasi and Mycobacterium fortinum are the non-tuberculosis mycobacteria. Okay, so moving on to the next question. Which of the following pairs are wrongly matched? Clostridium tetani affects with broken skin. Yes. Shigella infects the genitourinary tract. Yes. Salmonella affects the gastrointestinal tract. Okay. So yes, this is correct. Shigella doesn't affect the genitourinary tract. Okay. So moving ahead, which among the following are correctly matched? So rifampicin disrupt the membrane transport and energetics. Yes. Pyrazinamide inhibits bacterial DNA dependent RNA polymerases. Ethambutol inhibits the biosynthesis of cell wall arabinogalactane. Uh, So these two are wrong, and this is the correct answer. Okay, moving on to the next question. Which of the following does not belong to Mycobacterium outer membrane? So the correct answer is option 4. That means Lamb and Arabic electron are present as a part of the cell wall and the periplasm. Only mycolic acids are a part of the outer membrane of the bacteria. So this was the last question for today. Thank you guys for joining in. Let's see you next week.